Okay, so first, I'm going to be clicking on my phone quite a bit today just to check when it starts to feel like the clock in the back has completely stopped. So if it if it is legitimately 10:40 and I'm still blathering on, please put your hand up and let me know. I know students have a really hard time letting the professor know when when class is over. Usually it occurs not by putting the hand up, but like throwing all the stuff in the bag and getting ready to go. But just so you know, I'm trying to keep track of time in my head today. So I have academic advising appointments with my advisees today until about 12:30. So if you're one of the people who I've asked to come see me, between about 12:30 and 1:45 or so to give me some time to get ready for lab, I'll be in my office, and that's times that you can come today. Everybody should be looking at what we're doing in the book. Now is the time to be looking at the problems at the back of the chapter. I'll be posting sometime today. I think it's a journal and not an exam homework today, but an assignment today to look at. Given conversations that I've had with some students who have come to see me, when you're working let me say, my point in letting you work in groups or encouraging you to work in groups is so that you guys can get together and talk over everything, and so that if there's any difficulties, you'll have a number of other people to help you out. I've seen a few instances in which what's happening is each person takes a question, they do that question, they don't do any of the other questions, and everybody turns their questions into a single person and then puts it up on Blackboard. That's not the point, and then you're shortchanging yourself in not doing all the practice problems yourselves. So a lot of times the problems that occur on the exams are similar to the practice problems. That's why I write both, so that's why I give them. The thing to do, and you can even, if you want to do that approach, the way to do it would be to you can still have each person responsible for a question, but each person should try to do all the questions, and then you get together somewhere in a group and each person can present one of the questions, but you'll have the other 2 or 3 people in the group able to say, no, that's not what I think, when they do that. Or if you're the one presenting your question and you haven't thought about it correctly, then you have a safety net in other people who can help you out. So that's the point of what I've been trying to do there. If you're doing the problems at the back of the book, remember that they have in both the short answer problems at the beginning, and then later on when it's like the work it out problems, they are organized by subheading. So you can see section 10.1, 10.2, 10.3. So look at where we're at in the book and in the lecture, and do those questions, be doing those questions that match up where you're at as we go. Do not wait until the very end, or even waiting a week before the exam is cutting it too close. So right now you should be working on the stuff that we've already looked at. All right. So here's where we ended up last time. We're talking about Watson and Crick and their legitimate but Nobel Prize that they got, because there was some weird stuff going on here that shouldn't have happened. So, like I said, it was the X-ray diffraction pictures from Maurice Wilkins' lab, and specifically his colleague Rosalind Franklin, who actually got it to work, that allowed them to make their structure. So I talked about X-ray diffraction a little bit last time. Here's a picture of what it looks like. I said you can understand X-ray diffraction by imagining sunlight going through a prism, and it separates it into all the beams of light, because each of the wavelengths that come together to make white light has a different frequency, and the prism is able to separate them. So here you have in X-ray diffraction, you have a source of X-rays, and that goes through this lead screen. The reason why you want to do that is because the light source is, as you can see, producing, and it would be all around, the light source is producing different beams all around. And what happens when you pass it through the lead screen is it's only allowing one specific beam to go through. So it's only one spot. If you did not have the sample here, what would happen is you would get a giant black dot in the middle here. So what happens is in the old days, you would be exposing a piece of X-ray film here. And so if you did not have the sample, you would just get a giant black dot in the middle because the beam would go straight through and hit it. But instead what happens is the beam diffracts based on what's in the sample. So what's the sample? You have to take your biological material, and at the time biochemists had already done this for proteins where you make crystals out of them. And Rosalind Franklin was successful in crystallizing DNA. She was one of many scientists trying to crystallize DNA at the time, but she was the only one that got a crystal that made a diffraction pattern that allowed scientists who looked at it to figure out what was going on. So the difference was that her crystal had a higher hydration or amount of water that's in it versus the other ones, and we'll see different structures of DNA coming up, and the one that her crystal ended up being was bDNA, similar to this model that I have here. The less hydrated version was a wider helix and more compact, and so it didn't give a diffraction pattern that would have been decipherable to anybody. This is her actual photo 51 from her lab notebook. You can see here in the middle, it looks like a record. You guys have seen a record, maybe in a museum or something, where there's always, if you see these things, there's always a hole in the middle. Why is there a hole in the middle? Because there's a giant black dot in the middle, because even though you get this diffraction, most of the light comes straight through, like I said, and hits the middle, so that's just a black dot and it's meaningless. So I am not a structural biochemist, but I can tell you that when Watson and Crick saw this, because they had been in contact with other scientists like Linus Pauling, who had successfully crystallized proteins that form a helix, this cross forms, this little X that's formed here, is a dead giveaway that there is a helical structure. And they could figure out like the distance between the levels here, between the bases, based on the distance between these little bands here. And there is other information about this that I'm not able to interpret, but if this is interesting to you, just look up photo 51, and there will be a lot of there are multiple places online where you can see this exact picture, and everything about it is described. So here's the deal. Rosalind Franklin was the only one who got the good crystals that gave the good structure, but she hadn't figured out what was going on, because unlike Watson and Crick, she hadn't been out to talk to all the protein biochemists. She would have eventually figured it out, except that Maurice Wilkins just brought Watson and Crick, the old boys club, in and showed them her work without her knowledge or consent. And so she got screwed in multiple ways. First, they stole her work, and she could have been the one that did it. And second, She died of ovarian cancer at age 37 in 1958, and the Nobel Prize was in 1962. And except for one occasion in the last, I think it was like 5 years ago, they don't award Nobel Prizes to dead people. They did, it's happened once to my knowledge, and I can't remember if it was literature or something else, but the person who won it had died, but the committee didn't know they had died when they awarded the prize, so they let him have it. So they let him have it anyway. And this is tough because of this, right? This is why she died of ovarian cancer. She was working with an X-ray source, and they did not know about the bad news of radiation at that time so much. And she was, well, they did, you know, because of the atomic bombs and everything, but they thought this was a low enough and safe enough level. I mean, this is a lot more than you get at the dentist's office, and at the dentist's office they put that big thing on you and they go out of the room and all that stuff. So these exposures, I think this one, I'm not sure, but yeah, so 100 hours of exposure. So this thing was just running in the lab for days and days, and she was sitting there writing stuff and nuking her parts. She would have probably been screwed anyway, because Nobel Prizes are only able to be split 3 ways, and they would have given it to her boss. But really, she should have been able to figure it out, and then Franklin and Wilkins would have won the Nobel Prize, and not Watson, Crick, and Wilkins, who eventually got it. James Watson, while a brilliant scientist, is an all-around not so good guy for everything else. If you read this book, it is full of misogynistic stuff, like, you know, describing the way she dresses as not, you know, attractive and this and that. And if you read this book and see all the horrible stuff that's in it, keep in mind that's the cleaned up version, because this was shown to Francis Crick and other people who were fairly horrified at what Watson was just, you know, filter free coming out of his head, because he's a Nobel Prize winning, you know, whatever. And so this the cleaned up version. Also, he is the only scientist that I know of that has ever had, and so it came back to get him in the end. Justice prevails eventually. I think he's still alive, but he's the only scientist I know of who ever had to sell back his Nobel Prize medal. So like he sold it to get money. Because he was also, in addition to being misogynistic, he was also a terrific racist who said things that he thought to newspapers on multiple occasions. And he was chancellor of Cold Spring Harbor Lab, which is a famous molecular biology lab on Long Island in New York for a long time. But they, probably 3 or 4 years ago, the second time he said racist stuff just straight out, they're like, bye, and they took it away from him. So he was like the Chancellor Emeritus and all that stuff, and so they finally kicked him out, and he's fairly well in disgrace. So if you want to, and Crick is dead, but if you want to think well of one of these guys, think well of Francis Crick, who was both a brilliant theoretician and a brilliant scientist, and we'll see him come back again. And also, Francis Crick and his wife looked after Franklin in their home, I believe, while she was dying. So he's the good guy, not the good guy. If you want a good biography that's fair of Rosalind Franklin, you know, so everybody has their, you know, nobody's perfect, but nobody's perfect doesn't mean that you should have that bad stuff happen to you. So this is a fair biography that tells about her life in science and the short life that it was. Most libraries have this. I think ours does. It's a good one to read. All right, so After this was done, and I led on to this, we talked about bacteriophage having DNA as their genetic material and not RNA. There was no RNA in them at all. Some viruses have RNA for a genome, including some human viruses. So Frankel Conrad and Heinz Frankel Conrad and Bea Singer were working with another model organism virus, and this was tobacco mosaic virus. So what were the original, before Arabidopsis that I talked about, what were the original plant model organisms? They were tobacco and they were maize or corn. And the reason is because those are the valuable cash crops at the time. And so scientists were interested in learning how things work, and tobacco farmers were interested in learning how to make better tobacco, and corn farmers better corn. Higher yields, certain characteristics being better, and so both benefited. Tobacco mosaic virus is similar to bacteriophage in that if you take, let's just look down here for a second, if you take the protein that coats it and the RNA and throw them together in a test tube, they will spontaneously assemble to form the virus. Now it's not sticking out like this, this just shows what's in there, but the protein subunits sort of come together and circle around and around and around like a turret on a castle, right? What they did was, so tobacco mosaic virus, it infects tobacco leaves and it makes sort of a oily looking, you know, when you see oil in water and there's like that rainbow color, so that's what I think they do, that's the phenotype of it. And then the tobacco dies. And this was a problem because if you have your tobacco infected with tobacco mosaic viruses, those leaves aren't good and they have to be thrown away. All right, so there were two different types of tobacco mosaic virus. No, they are not red and blue in real life, but they're just shown this way. So there's each one of them has different proteins and different RNA, and they can be told apart. So what they did was they were able to dissociate the RNA from the proteins, and then they mixed them up. So they were able to combine the type A RNA with the type B protein, and to do the reciprocal, the type A protein with the type B RNA. And they came together in a test tube to make a virus that was infectious. Then they took these hybrid virus particles and infected tobacco leaves with them. After the tobacco leaves were infected, they got the disease. Out the other end came replicated virus particles that could infect something else. But the RNA type, here the red and the blue, turned out to be what the whole virus was later. So RNA is the genetic material because the virus that comes out after infection matches the RNA type, not the protein type. So this experiment did not involve labeling the protein and the RNA like the Waring blender experiment did, but they could tell the RNA and the protein apart, and by infecting with this one, for example, on the left, you have that type A RNA and type B protein, but the type A RNA codes for making more type A RNA and the type A protein that goes on there. So this showed that in some viruses that RNA can actually be the genetic material. So it was like, what? Because the scientists looked at these viruses and found out that there was no DNA in them. So in bacteria, in the Waring blender experiment, it was easy to say DNA is the genetic material here because the bacteriophage only had DNA and the protein coats, the little capsids there. Here, it's the same thing, but with RNA only. There's no DNA in these guys at all. All right, so put on your biochemistry helmets because we've got to do it. See that, but it's something that's actually, you can see how things work, right? All right, so as you may know from introductory biology, many of the all of the large macromolecules are just polymers. So you have polymers of sugar, you have polymers of nucleic acids, which are going to be DNA, you have polymers of amino acids that are going to be proteins. Simple building blocks that come together in chains, sort of like a bunch of train cars that are connected. So we're going to examine the components of a nucleotide. It's one of the building blocks of either DNA or RNA. So the first thing is a pentose sugar. Why? Pent means 5, right? So pentose is 1, 2, 3, 4, 5 carbon sugar. And there's an oxygen in there. 4 of the carbons are within the ring. 1 of them is outside the ring, and that's how you keep track of them. So you're going to have to know the 1' through 5' numbers here, because we're going to see that it's important for how the chains come together. So you can either remember that the 1' is within the ring, or you can remember that the 5' is outside the ring. As we'll see, that nucleic acids, nucleotides form chains by having bonds at the 5' and 3' ends. If you remember that, then you'll know that this one and this one, if you look at a structure, have to be, one of them has to be the 5' and one has to be the 3'. And the 3' can't be outside the ring because it wouldn't make sense because the numbering goes in order. All right, so there's a difference between the sugars at the 2' carbon. So in ribose, you have a hydroxyl group at the 2' carbon. That is gone here. Deoxyribose gets its name because the deoxy part means that the hydroxyl group is gone from the 2' carbon. That is very important for 2 reasons. The first reason, when we talk about nucleic acid polymerization in DNA replication in chapter 12, is how you make a DNA chain and don't get RNA nucleotides in there. Believe it or not, that tiny little hydroxyl there causes steric hindrance and keeps RNA nucleotides from going into DNA chains. And then when we talk about splicing, so removing introns from pre-messenger RNAs even further on in the future, without this hydroxyl there in messenger RNAs, splicing would be impossible. All right. So, like I said, the phosphates in the chain are going to be attached to the 5' and 3' carbons here. Talked about that. And then the base is attached at the 1' carbon. So in this model, the bases are in the middle, and so you can see this would be the 1' carbon in the ring, and here's the base. Where are we at? This is the 1' carbon here, and this is the base. Right? We call them nitrogenous bases because they contain nitrogen. So in this model, the carbons are black, the phosphorus is purple, nitrogen is blue, hydrogen is white, and oxygen is going to be red. All right, so here are the bases, and there are 5 of them: A, G, and C, or adenine, guanine, and cytosine, are used for both DNA and RNA. But where DNA uses thymine, RNA uses uracil, and they differ only by this methyl group, which is here and not there. You do not need to know the numbering for these because it's not important for anything that we're going to do in this class. You do need to recognize what's a purine and what's a pyrimidine. Purines have 2 rings, pyrimidines only have 1. I will not ask you to draw these structures, but if I show you one of them, you need to be able to say what it is. Fair enough? The way you tell what's a purine and what's a pyrimidine is with this very nice mnemonic device. So purine starts with PU, pyrimidine starts with PY. That sounds like pie, right? Before you eat the pie, you have to cut it. And the pyrimidines are C, U, and T. Isn't that cute? If you have a better one, let me know. But that seems to work for most people. Also, the world's most giant phosphate. So this is what's actually going to hold the sugars together. And this is why in the Waring blender experiment you were able to label DNA to such a high degree, because they used the 32 isotope of phosphorus, so P32. Every nucleotide is joined to the next one by a phosphate here. So in order to make a nucleotide, you have to first make a nucleoside. So a nucleoside is the base plus the sugar. And this bond is way over exaggerated to, you know, there's no way that bond is that long. It's just to show that the base is sticking up there. But now you can see that the base is attached to the 1' carbon in all those. So that's another way to remember it. The 5' and 3' attach one sugar to the next one. The base is always attached to the 1' carbon. And then you can count 1, 2, 3, 4, 5. The oxygen is not a carbon, so it is not numbered. Going from a nucleoside to a nucleotide just involves adding one or more phosphates to the 5' end. And there we go. So these are nucleotide monophosphates because there's only one phosphate here. So deoxythymidine 5' monophosphate because the phosphate is at the 5' end. So adenine, guanine, cytosine, and thymine refer to the bases if they're just floating around by themselves. If you add the base to the sugar, then these suffixes change. So adenine and guanine, the purines, the ene changes to ocene here. And in the pyrimidines, thymine, cytidine, and uracil, or thymine, cytosine, and uracil change to idine. The uridine is not shown here, but like thymine changes to thymidine, cytosine changes to cytidine. The difference in those names means that you have that sugar attached to the bases there. Here's the phosphates. DNA and RNA are built from nucleotide triphosphate. So what's this mean? DTMP. The D means deoxy. So that if you see a little lowercase d, that means you're talking about DNA bases. Deoxythymidine, that tells what base, and then monophosphate. So DNA is built from nucleotide triphosphate, so it would be DTTP, DCTP, DATP, and DGTP. So as we'll see as soon as I get my batteries changed here, I can just go to the next slide right through this. You can see how they go together there. You can also see how there is a difference in polarity between the two strands. So back in business. So while on the left-hand strand on this picture, the 5' end is facing up, on the right-hand one, the 5' end is facing down. So figuring this out, that the two backbone strands had opposite polarity, was one of the big things that Watson and Crick did. Their two big things figuring it out was, or three things was, one, looking at Rosalind Franklin's picture and figuring out it was a helix. Two, figuring out that to fit it together, you had to have the strands going in what's called an anti-parallel orientation. So they're running parallel to one another, but one strand is in one orientation, the other strand is in the opposite, and then figuring out how the bases fit together using those cardboard models that they had. This is just this if you were to unwind it and flatten it out so you can look at it. Right? So there are 4 types of bonds involved in holding this together. The backbone here, like this, the backbone here, the chains are held together by strong covalent bonds. These are called phosphodiester bonds because an ester is a bond involving an oxygen, and so there's a phosphodiester. There's a phosphate in the middle and an oxygen on either side linking to the carbons. So here is a phosphate that I'll get you in a second. So look up here for just a second. Here is a phosphate, and then over here and over here are the two oxygens. So a phosphodiester bond holding it together. These are strong covalent bonds, sharing of electrons, very stable. Yes? Yes. Oh, so the DNA is built from nucleotide triphosphates. And what I failed to mention is you can see here there's only one phosphate between them. The other two, we don't get to this until chapter 12 when we talk about DNA synthesis. The other two phosphate, or actually we do. No, we talk about it. Nope, not until chapter 12. I thought I had something in here about it. When a new nucleotide comes in to elongate the chain, 2 of the phosphates are removed, and there's a huge energy reduction in that step, because when the 2 phosphates come off and together and then dissociate into single phosphates, it's a huge energy drop that makes sure that the reaction goes in one direction. So the energy for putting these together, as we'll see when we get to that chapter, comes from it being a triphosphate. The cell has to expend a lot of ATP to move those phosphates on there and make them high energy. And then when they're put in like this into the chain, this is a lower energy state, and that's what drives the reaction. Yes? What? So, pretend this isn't here, right? When you're going to add to this chain, the DNA polymerase is going to be sitting over here, and in comes a CTP with 3 phosphates. It will be a C because C pairs with G and A pairs with T. And as it comes in here, the enzyme causes removal of 2 phosphates that just float off into space as this bond is made. And that is a slight energy reduction, but when those 2 phosphates, which is called a pyrophosphate, when the 2 phosphates together break apart into single phosphates, that's a huge drop in energy, which makes sure that this reaction wants to drive forward. So as the nucleotide triphosphate is added to the chain, 2 phosphates come off, and this bond is made right here. So this bond is already there because in the nucleotide triphosphate, you would have the 5' carbon with 3 phosphates, and the other 2 that would be over here pop off. So this bond is created. This was originally hooked to the other 2 phosphates, and you exchange this bond to the carbon there for the bond to the other phosphate. Yep. Yes, you would have started with DCTP. Now you do not have DCMP because it's part of the chain now. So it's part of a DNA chain. It loses yeah, right. It would be this, but technically it gets married into the chain, and so it's not its own separate unit anymore. Good? And what came from Erwin Chargaff's rules, where he found that the amount of A is always equal to the amount of T and the amount of G is always equal to the amount of C, led Watson and Crick to propose correctly that A's and T's pair and G's and C's pair. So you can have a DNA molecule that has more A's and T's versus G's and C's, but the A's and T's always have to be equal because they're always paired, and likewise for the G's and C's. All right, so here, let's go back. Bond type number 1, strong covalent bonds holding the chains together, because you don't want them to just fly apart. That wouldn't be good. However, these are the bonds that get ripped. Remember in chapter 8, we talked about if you have the inversion heterozygote, and with the paracentric inversion you get that dicentric bridge and the chromosomes are pulling apart. It's one of these covalent bonds that gets ripped because it's not strong enough to resist that kind of force. All right, then the anti-parallel strands are held together by weaker hydrogen bonds. So hydrogen bonds are formed by partial positive and partial negative charges. Does anybody remember, I'm sorry to do this to you, does anybody remember from chemistry what electronegativity is? It's the an atom's propensity to attract electrons, or the affinity for a specific nucleus for the electrons that surround it. And it's different. Usually the bigger the atom, the stronger it pulls in on its electrons. So there are covalent bonds. They're sharing electrons, right? But just like a lot of sharing arrangements, it's not always equal. So for example, look at this nitrogen. The nitrogen has a lot higher electronegativity than the hydrogen. Not enough to steal the electron from the hydrogen, leaving just a hydrogen ion with no electron, but these two share the 2 electrons that complete the first shell on the hydrogen there. But because the nitrogen is much more electronegative than the hydrogen is, on average, the electrons are more close to the nitrogen than to the hydrogen. Meaning that the nitrogen has a slightly negative charge, partial negative charge it's called, and the hydrogen will have a partial positive charge. So this hydrogen has a partial positive charge. Let's look over here. This nitrogen over here is going to be more electronegative than carbon. And so even though it's sharing, there's resonance around here, around the ring. So even though that exists, there's the two nitrogens here in the ring with the carbons will have the electrons around them slightly more often than not, leading to this being a slight negative charge. And in this case, this slight negative charge attracts the slight positive charge there, and it's sort of like magnets coming together. This is a very weak force, but if you have enough of them, like through a long DNA molecule, it's plenty to hold it together. And because the hydrogen bonds form specifically between A's and T's and also between G's and C's, it allows the anti-parallel strands to line up correctly. Let's look at this guy. Here's an oxygen. So oxygen also beats carbon. So there's a double bond here on between the carbon and the oxygen, but the shared electrons are around the oxygen slightly more often than they are around the carbon, giving the oxygen a partial negative charge. Here's a hydrogen with a nitrogen, and just like this arrangement, the nitrogen has a much stronger electronegativity, and so even though there's sharing of electrons here, they're around the nitrogen more often, meaning the hydrogen is slightly positive. So a positive here and a negative there. In the hydrogen bonds that form between DNA strands, the hydrogens are always going to have a slight positive charge, and they're always going to interact with either a nitrogen or an oxygen that has a partial negative charge. Make sense? There are two hydrogen bonds that form between A's and T's, and 3 that forms between G's and C's. If you look at this model, which I will have in the lab today, so here's a GC pair. You can tell this is the G because it's the purine, this is the C because it's the pyrimidine, and then here are the bonds between them. You can see that this little connector, which represent a bond, this one, and this one are slightly these 3 are the ones that are slightly longer, and those represent the hydrogen bonds. It's not a real bond like the other covalent bonds, but the only way they had to show them here on this model was to give the slightly longer little plastic connectors. Because AT pairs have 2 hydrogen bonds and GC have 3 hydrogen bonds, the higher the GC component of a given piece of DNA, the stronger the 2 strands hold together. The double helix of DNA can be separated into single strands if you break the hydrogen bonds. Because they're not covalent bonds, you're not ripping the structure apart. They can come apart and go back together, come apart and go back together freely. And that's important because you have to be able to separate the double-stranded DNA into single strand temporarily for things like transcription, making an RNA copy, or if there's DNA damage, to repair one of the 2 strands. Sometimes you want to separate them permanently, like when you're going to make new DNA during replication. You'll separate the strands, and as we'll see, each one gets a copy attached to it. Separating the strands is termed melting because one of the ways that you could do it artificially is by raising the temperature. So if you take DNA, put it in a little tube, stick it in a water bath. As the water bath gets close to boiling, like about 95 degrees, that added heat and more chaos makes it so that the hydrogen bonds are not able to hold the 2 strands together anymore, and they can unwind and come apart. But If you remove the heat and cool the two strands very slowly, they will have time so that all the G's can line up with all the C's and come back together. So renaturing, reannealing, hybridization, depending on the situation. The hydrogen bonds can be reformed. So renaturation or reannealing is when the original DNA molecule is going to come back together. Hybridization occurs when you mix DNA molecules or DNA and RNA molecules, and you have two strands coming back together that were not originally part of the same two strands. In addition to heat, you can also cause denaturing through high pH. When we do the experiment in the lab where we transform the cells with DNA next week, the following week we're going to be making or harvesting DNA from those cells. In order to harvest the DNA from those cells, we're going to have to break them open. The way we do that is exposing them to a high pH. So briefly in that procedure, the DNA is going to denature like this, but it's going to come back together and everything's going to be fine because we'll lower the pH back to normal and it'll be able to snap back together. The higher the GC amount, the more heat is required to cause them to dissociate. So each given piece of DNA will have a melting temperature. If you have a really long piece of DNA, it's going to be, up fairly high. The length of the DNA raises the melting temperature as well because you have more hydrogen bonds. But even with a long piece of DNA, you have differences in the temperature at which the strands separate based on the GC content. So one way before complete genome sequencing was invented to determine the GC content of organisms was to figure out at what temperature the DNA would come apart. And the lower the temperature it would come apart, the higher the AT concentration in those DNA molecules. All right, so we talked about DNA so far. RNA molecule is usually single-stranded, as we'll see later on. It can base pair just like DNA can, but generally in the wild it is so to speak, it is single-stranded. Instead of thymine, it uses uracil, and instead of deoxyribose, it uses a ribose sugar. See here on the 2' carbon, there's that hydroxyl group, which is such a small substituent, but really it turns out to matter. As I said, this is critical because when you're adding, when we talk in chapter 12, when you're adding new nucleotides to a chain, this is what prevents RNA nucleotides from being added to a DNA chain. They don't fit into the active center of the DNA polymerase. And I mentioned about the processing as well. The double helix is the stable part of how the molecule holds together. Surprisingly, DNA is a very stable molecule. So when I send DNA off for sequencing, it doesn't require, being frozen or any kind of special handling. I just put it in tubes and send it off in the regular, like 2-day mail or whatever. When I'm storing it in the lab, of course, I store it in a freezer at like -20 degrees because it is an acid, and so it can act on itself and break itself up over time. However, over a couple of days, that's not too much. So why is this stable? Believe it or not, again, the hydroxyl on the sugar makes it more reactive for RNA. DNA is not. So working with DNA is just fine. You guys will do it and everything will be fine. But if you're working with RNA, you got to wear gloves, you got a beard, you got to cover that. I mean, anything, you got to not be over the tubes because your skin is crawling with RNase enzymes. And it makes sense because you don't want viruses that have RNA to get into you. So you just look at RNA wrong and it dissociates and it comes apart, right? The double helix sequesters the reactive base side groups inside. So look, here's what this looks like, but if you look down the barrel of it, except for at the ends, all the bases are safely tucked inside, and a lot of the little side groups are away from things in the cell that can cause them trouble. Remember, I just said that RNA is very sensitive. In some techniques, I use a technique called RNA interference, which allows DNA to be degraded based on providing an RNA that matches the sequence. The important thing there is that I introduce double-stranded RNA. So while single-stranded RNA is very easy to kill and ruin your experiment, as soon as I take two single-stranded RNAs that match so that they're anti-parallel and can come together to make a double-stranded molecule, once that happens, literally the gloves come off. Because once you are a double-stranded RNA, it's fine. You can handle it just the same as DNA because the side groups are now inside the cell or inside the helix or even the molecule, and the RNase enzymes can't access it as easily to chop them up. So we mentioned two kinds of bonds so far. The covalent bonds that hold the chains together, very strong. The weaker hydrogen bonds that hold one chain to another. And there's also, chemistry alert again, van der Waals forces, right? So you thought you were done with all this stuff. Van der Waals forces are involved with the stacking of the bases. So you get interaction of one base with the one directly above and directly below it. These bases are planar molecules, so they're flat and they can interact. So the stacking of the bases together, this rung in the ladder interacting with this one and so forth, has a stabilizing effect. That's even weaker than hydrogen bonding, but when you take into account the whole thing here, it adds up and leads to the stability there. The fourth kind of interaction is ionic interactions, and so all the phosphates here are having a negative charge on one of the oxygens. One oxygen, as you saw, is double bonded, the other one has a negative charge, and so these will interact with positive ions of salts in the buffer. All right, so how to change this. Between last semester and this semester, we're going from the FONS test to the Facebook test because nobody knows who the FONS is anymore. So we already talked about this earlier in class, right? So you guys remember how to tell that it's a double helix. So this is a right-handed double helix. So if you stick out your right hand in front of you, point it, with the thumb pointing up, you can see that this is twisting in this way around in this direction from the bottom to the top, which is the same as going from the palm of your hand to your fingertips in that curling way. So bDNA, which is this guy, is a right-handed helix. If you would have left-handed, it would be going the other way. And I'll have this in the lab so you can look at it. All right, so DNA has to interact with a lot of different proteins. The proteins that are going to make new copies of it, but more importantly, proteins that interact with the promoter regions. So those switch regions that show you where the genes should be turned on and turned off. If you look here, let's do, where's a good spot? Right here. Can you see how, and this is not a perfect model, but can you see how these, oh, here's better, right? Can you see how these two are very close together, right? And then there's a bigger distance here? Because they're coiling around each other, and you can look at it, come and look at it in the lab, it'll help you understand. Because of the way they're coiling and the way the bases interact, everywhere, if you go around this side, look, this is always going to be small, and this is always going to be big up here, right? You can start at the bottom here. This is the, there we go. This is small and this is big. This is small and that would be big. They alternate. But look, if this is small on this side, right here, on the other side is the bigger area. These are the major and minor grooves, so to speak. Proteins interact with the major groove versus the minor groove because it's bigger area. So if you look at the distance that forms the major groove here, if you take a protein and make an alpha helix out of it, so a little coil out of it, that alpha helix is just big enough so that it can lay right down into the major groove, and then amino acid side chains sticking out of that helix can interact with the bases there. And that's how proteins can read the nucleotide sequence and interact with them. When the second to last week in lab, we're going to be using restriction endonucleases. So these are enzymes that recognize a specific 6 base pair DNA sequence and then cut there. How do they do that? They interact with the side chains here. Common issue students are having is like, okay, so these bases have a major groove and these have a minor groove, so that means that these can't be recognized at all. This is not true, because on the back side of the major groove, sort of look through here, you can see the minor groove is over there on the back. And on the back side of this minor groove, you can imagine the yellow ribbon wrapping around. So it's this green ribbon wrapping around and the yellow ribbon wrapping around and around. So the major groove for this part is on the back side of the screen behind you. So every bit of the DNA sequence has a major groove. It's just the major groove is on one side and the minor groove is on the other. And that's how the protein interacts. So now we're going to look at how Rosalind Franklin succeeded where others would have probably gotten it eventually but didn't. So if you look at the A and B forms of DNA, you can see that these are both a right-handed helix. But the A form is compacted, it's squat down, and so it's like if you wound this tighter. There would be more bases per turn, and the shape of the molecule changes. There's only 75% water, so like I said earlier, that's what lower hydration means. This is what everybody else was making, and their crystals weren't working for X-ray diffraction. Rosalind Franklin's big breakthrough was that she made the higher hydrated form, that is the longer, skinnier form that did work very nicely for X-ray diffraction and gave that really good picture. So if her work wouldn't have been shared, it's not guaranteed that she would have won, but she might have had a chance. So she might have been able to figure out what her good data meant because the information was out there, or somebody else could have made bDNA who knew what it meant, who could instantly recognize it. Who knows what would have happened, but we will never know because they stole her stuff. The third form is what's called Z-DNA, so it's zigzag. Like you can see that the bases sort of zigzag back and forth across the helix. Z-DNA only exists where there's alternating purine and pyrimidine bases, like GC, 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 right, over and over and over again in a strand. And therefore it would be like AT, AT, AT on the other strand. It is called zigzag DNA. It's longer, skinnier, and it is the only one of the three that is a left-hand turn. This can be made in the laboratory, but as far as I know, we still don't know if this ever exists even in DNA with these repeats in real life. The B form of DNA is closest to what DNA actually looks like in cells. This is 10 bases per turn. This one is 11. Really, it's sort of a hybrid between the two. So in cells, it's about 10.5 bases per turn is what it ends up being. The best estimate that we have. All right. So I've mentioned the central dogma a little bit before. I'm going to show this version of this diagram over and over again for the rest of the class to show you here's where we're at, here's what we've already seen, here's where we're going. So as soon as, or very soon after the solution of the structure of DNA, Watson and Crick proposed what they called the central dogma of DNA. So this is how it is. The dogma is like, this is how it is. And there were some changes to that over the years. So DNA is maintained by replication, so you can make more DNA. Phenotype is encoded by DNA, but in order to see the phenotype, certain pieces of the DNA, genes, have to be transcribed into RNA. So transcription was chosen because transcribing something just means making a copy of it. Yes, RNA has different bases, and yes, RNA has different sugars, and it has uracil instead of thymine, but it's sort of like you're sort of like copying out of a book, right? So the scribes before the Gutenberg invented the printing press would have to very carefully copy from one book to another, just like DNA or RNA polymerase will use DNA as a template and copy RNA. Then, in order to show the phenotype, RNA has to be translated into protein, and this is called translation because it is literally changing language. You're changing from the language of nucleic acids with 4 bases to the language of amino acids that make up proteins with 20 bases. And during the 1960s, at the same time, the United States was racing the Russians in the space program to see who could get to the moon first. There was another just as major scientific program going on in which we were trying to figure out how the 4 bases in RNA can code for 20 different bases in proteins. And there were a lot of really good experiments that were done during that time. And that's it to start with, like the Watson and Crick original central dogma. So how is this regulated? We're going to talk about this too. So transcription is very highly regulated. And we're going to see it's regulated both by the presence of transcription factors or proteins that are going to read the promoter and say start transcribing here. They have to be present at the time and place where transcription of a particular gene is going to happen. We're also going to see how the DNA is packed into nucleosomes with histones is also important. So it used to be thought that transcription was only controlled by whether these transcription factors were present, but now we're showing that how the DNA is packaged is just as important or more important for controlling that, because if you have DNA very tightly packaged, it can't be transcribed into RNA even if the transcription factors are present, because they can't access the bases they need to. In many cases, translation is regulated too. So the cell can have a bunch of RNA laying around, but there are special mechanisms by which the ribosome interaction with that is regulated so that translation can only occur at certain times. The reason for this is because oftentimes a cell will want to make a whole bunch of protein as quickly as possible. And when you start up here, you have to get transcription going and then wait for that to make enough molecules, because there's only going to be 2 copies of each gene in the cell. And then the ribosomes can start translating that into protein. If you accumulate a bunch of RNA but don't start translating it, then you can, when the signal is given, the ribosomes can latch on to all these molecules of RNA and go for it. Don't talk about it here, but in cell biology, if you take it, and I recommend you do, degradation of proteins by the 26S proteasome, and also regulation of proteins by usually phosphorylation and dephosphorylation. So you can have proteins sitting around, and they can be active or inactive depending on their phosphorylation status, in which phosphatases and kinases take off or add phosphates. In some cases, the protein with the phosphate on is active, and in other cases, the protein with the phosphate on is inactive. Not thought of until the late 1990s, so reported in 1998, I believe, the phenomenon of RNA interference. So this will allow degradation of RNAs, and this was a huge breakthrough, but not originally envisioned by Watson and Crick. Also not envisioned by Watson and Crick was this phenomenon, reverse transcription. So because RNA and DNA are both nucleotides, it should be possible, just like you can take a DNA sequence and make an RNA sequence by matching up the complementary bases, we call it, you should be able to go backwards. And originally Watson and Crick thought that, they said it was possible, but it doesn't exist. But then in some viruses, it was found that viruses with an RNA genome have to convert that back to DNA, and so there are enzymes called reverse transcriptases that can take RNA and make it go back to DNA. Lately, we've also found that RNA can be directly replicated without changing it to DNA as well. And in many of these cases, my postdoc boss liked to say, anytime you make a discovery that in some way modifies the knowledge around the central dogma, then you get a Nobel Prize for it. And it's true in a lot of cases. So discovery of reverse transcription was a Nobel Prize. Discovery of RNA interference was a Nobel Prize. Discovery of how RNA codes for protein was a Nobel Prize. I don't know if that was or not. I'll have to look it up, because one of the guys who should have got it for that didn't get it until later. Anyway, I think I'll stop there for today. Go ahead and read the rest of the chapter. I've got the next chapter in there, so go ahead and print out the handouts if you want. If you're in the lab today, I'll see you in lab. Remember, if you're one of the people I asked to come see me to talk about study strategy, I will be available starting at about 12:30 today after my advising appointments are over.